All right. Okay, so I will be talking about sequencing analysis, and I think I'm the only sequencing talk from the primer point of view. So I'll try and give a little bit of a melange of the concepts behind sequencing, where the data come from, some of the study design issues, and then try and touch on some of the analytic issues and approaches. I mean, it's a kind of big space, and you could spend an entire primer semester on sequencing alone, but we'll just dive right in and see how we go. So really, I'll try and talk about all of these things. There are bits and pieces of the talk that I can drop out in the event that I run a little too slowly or whatnot. But really, the important things that I hope that you'll take away from this session this morning are some of the core concepts of where the data originate, some of the testing approaches that are currently available in the community, and then some of the other ways that we're thinking about leveraging sequencing technology to understand a little bit more about the genetic architecture of both Mendelian and complex traits. So why sequencing? Well, there's a lot of exciting potential here. This is a slide I cribbed from Mark Daly, and it's just a kind of series of papers that we can walk through, each of which represents the discovery of a kind of causal variant or causal gene for some very severely deleterious diseases, so things like Miller syndrome, or this congenital chloride diarrhea, or very unusual IBD, or secondary lung carcinoma. I mean, there are a lot of these very early onset, very, very deleterious diseases that are really, really nasty, that we've suspected for a long time are probably genetic in nature, but haven't really had the technology or the ability to ask questions directly about all of the variation in, say, the coding regions to try and uncover whether or not we might actually learn something a little bit more about the biology aside from the clinical presentation. And that really is the kind of, first major contribution of sequencing, of exome sequencing and genome sequencing, I'd say at this point in time, is really getting an insight into these very rare, very deleterious, very, very nasty diseases. Now, these are, the success stories, but it's not all successes. If you come around to ATGU and come to clinical sequencing hour, you can see, diseases just as similarly severe as some of these presentations. And we're not batting 1,000. It's not like we just run sequencing and we immediately get the answer because we identify the very nasty deleterious mutation. And there are a couple of reasons for that, and probably the most natural and intuitive one to think about is the fact that we have a huge amount of background variation to contend with in the human population. And this really stymies not only our kind of Mendelian rare nasty disease experimental work that we're trying to uncover the biology for, but also the common complex disease work that we're trying to uncover the biology for. So, your things like autism or schizophrenia or heart attack or type 2 diabetes or all of those things. So really this, background rate of variation, if we think about an individual genome, we'll have, maybe somewhere on the order of 80 point nonsense mutations in your genome. And that's, it's kind of on average across the entire population. That's a lot of, mutations to kind of juggle that it's not clear that all of them are going to be clearly causing nasty disease. Like now most of these, or a great many of these, are actually common in the population, and so we can kind of rule them out as causing some of the most deleterious, nasty, rare diseases. But in terms of really, understanding the impact of these mutations, we're going to require larger sample sizes, and that'll be a kind of common theme throughout the talk. So I'm going to spend a little bit of time trying to talk about where the data come from, because really, I think sequencing is kind of like a black box, and it's a little bit more complicated than, say, array-based genotyping. I could do an array-based genotyping cartoon in one slide, and that would give you the general idea, but that's not really how it works for sequencing. So the kind of first major divide that exists in the sequencing world is the extent to which we are trying to capture variation. So there are really two places that we go hunting. We go hunting across the genome at large, completely try and get everything as best we can. And then we also kind of drill down explicitly into the exome itself. Now, the exome represents 1.5% of the genome, more or less. It's basically all of the exons, all of the transcribed and then translated bits of the genome is the general idea. And the motivation for targeting this part of the genome is kind of twofold. One, it's enriched for strong biological consequences and impacts The selective depression of variation is most pronounced in the coding region of the human genome in contrast to anywhere you look. Encode regions, whatever, doesn't matter. The most selected place in the genome is still the coding regions. That is just the way it is. And then the second thing is, for the things that we have understood and identified thus far, for at least certainly with Mendelian disease, a lot of those mutations really are captured in the coding region. And that's, there's a little bit of an unfair advantage to the coding region because it's also more interpretable. So this interpretability, this ability to understand exactly what a mutation is likely to do, have a very good shot at kind of parsing its biological consequences, really improves our understanding and kind of at the end of the day, we do genetics not because we like genetics. I mean, we do like genetics, but we do genetics to try and uncover biology at some level, and being able to actually understand what the variation is doing and have a pretty good prediction about the functional consequences really does improve our interpretability of the experiments that we're doing. Now, this requires a step called library construction. I'm a statistical geneticist. I am not in any way, shape, or form a molecular biologist, but I will give you a cartoon example of what that is in the kind of fumbling way that I can best manage. So that stands in contrast to the genome. Now, when we try and look at the genome, we have a kind of much bigger space to deal with, okay? Because the genome's a very, very big place, 3.3 billion bases. And as a consequence, we tend to go at a lower depth. Okay, now I need to take just a moment to define this term depth or coverage or depth of coverage. For the first 2 or 3 months when I started working in the sequencing space, people were like, depth, coverage, and I was like, what the hell are they talking about? I don't understand. This is all very confusing. And really, the core piece of information from the sequencing data set is a string of bases. So we get a string of bases as the read-off from the machine. And I've got a lot of pictures trying to show this. But the depth is really the idea of the number of times that you can align a string of bases to a position in the genome. So it is basically the number of data points you have for identifying whether there's a variant there or not. So this concept of depth is really important because, as you might imagine, the deeper you go, the more data you've generated, but at the same time, the deeper you go, the ability to identify variation and those sorts of things, that improves, but it also increases the cost of the experiment and the storage space. So these data sets get very, very, very big. So to give you a kind of idea, the exome, we do deeper coverage because we want to capture nearly all of the variation that's identified, that is potentially there in the exome. And that'll be somewhere on the kind of 18 to 20 gigabases gigabytes for an individual, in their BAM file, in their kind of stored output. For a genome at 4x, that's about the same. It's about the same, but it gets much, much larger very, very quickly. It's a little bit more for a genome, but if you try and start to go 30x or 60x, which means you're really trying to capture all of the variation within an individual at all places across the genome, you're talking about a very, very large amount of data, on the orders of hundreds of gigabytes for a single individual's genome in terms of computer storage. So there are computational challenges associated with this activity as well, as you might imagine. Okay, so here's the cartoon, the statistical geneticist guide to how hybrid selection works, which is really how we target a specific subset of the genome to try and identify the variation in that subset very well. And really what we've got are two kind of parallel activities. So the first parallel activity is this microarray bait development activity. And basically what we're doing is we're generating this pool of oligonucleotides. So what that means is we're just creating a pool of strings of bases, of bits of DNA of specific strings, specific sequences. And then we PCR amplify those. So we have a little template and then we make many, many copies of those templates. And then we do this transcription for a single strand RNA bait. So the idea is that this RNA is what we're going to try and have bind to the DNA so that we can pull the DNA down. At the same time, what we're doing is we're taking our genomic DNA, we're randomly shearing the DNA. What I mean by randomly shearing the DNA is really I'm randomly breaking up the DNA into little bits and pieces. And we can do that by doing something called sonication, which basically makes the DNA vibrate at a certain frequency. Because it's a coil, when it vibrates, resonance kicks in, and then it breaks apart. Really lovely physics if you kind of want to think about the biophysics of the activity. We then ligate an adapter. All that means is we stick an adapter on the end, and then we PCR amplify. So we make lots and lots and lots of copies, okay? So then your idea is to have this bait and have this pond. So the bait is where we're going to go, like, that's what we're going to use to go fishing. So we're going to take that bait, and that's going to be, kind of identical to a sequence in the human genome. And we're going to try and lay things down to grab that particular chunk of DNA using this bait. We then anneal this to a pond mate, so that's this part here annealing. We then capture it on the magnetic bead here, so that's the kind of solid state technology. We PCR amplify, and then we analyze on a next generation sequencing machine. So that's the kind of cartoon overview. You can go and read Gernicki et al. In 2009 that really kind of identified the kind of core process associated with this particular technology. Now, each of these, technologies gives us a sequence of bases. So we get a string of bases, maybe 76, maybe 101, maybe much longer than that, maybe shorter than that. And we get many, many, many of these strings of bases. Well, now what do we do? So this gives us our raw short reads. And there are a variety of technologies that are used. Here at the Broad, most of the technology we tend to use is the Illumina Solexa technology. So Illumina bought Solexa a few years ago and then kind of ramped up the technological capacity and the kind of throughput. And so that's what we're using currently. But again, sequencing is a changing sphere. So I'm sure that in a year, this talk will be outdated in terms of some of the things that I tell you. But at a core level, this paradigm isn't going to change in the near future, I don't think, which is you get these raw short reads, so these strings of bases. And what do you do with them? Well, you try and align them to the human genome reference. So we have this reference genome, and that reference genome really, really defines so much of this activity. Without the Human Genome Project, sequencing as we know here would be effectively impossible. It would be a much more computationally challenging task. And if you want to learn about how computationally challenging a task it would be, you can go talk to the people in assembly, which is, stitching together genomes. Without having a reference, terrible work, really hard. So we've got this mapping and alignment, and we're trying to kind of match up where things go. We then, once we've mapped these reads, so we've kind of plunked down the reads on the genome, we're like, you read, you go here, so on and so forth. We then have to do a kind of quality calibration and annotation of the read information itself. So these technologies, all of these technologies give you a sense of the quality of the individual base call at each base call that is made, okay? So they're like shining lasers on things and zapping things, and those intensities, they, permute through their, kind of closed corporate algorithm that then defines this quality score. Well, those quality scores need a little bit of help because there's a lot that's going on technologically and kind of data quality-wise that isn't being captured by their algorithm. So we have to kind of fix that, and I'll talk you through how that works. And then you identify genetic variation. And then once you've done that, you can actually start analyzing your data. So it's really a very, very long and complicated process. There is one other thing to flag. Most technology now is paired end sequencing. This is also confusing. It's like, what the hell is that? Well, really, when you do that sonication thing, you do the break it down and all that kind of stuff, the idea is that you actually try and sequence from both ends of the DNA fragment that you've isolated. Okay, so you're going to try and go from one way, from positive to negative and from negative to positive. And the reason that you do that is twofold, but the kind of most useful part of it is that it really improves the quality of the data because you're going to get information from both directions, and there are certain errors that are more common given sequence motifs of one side versus the other. It also helps a little bit with things like phasing and calling haplotypes and those sorts of things. So that's the general, that's one other additional little tweak to be aware of. Okay. Now we're going to talk about a series of kind of core basic concepts when evaluating sequencing data. So coverage is the number of reads at the position. Coverage is king. The more coverage you have, the better you will be able to identify variation, the better you will be able to QC your data and understand the properties of the variation that you've identified. With coverage, it's the poison chalice of more and more computational space and time and all of those things to kind of grapple with. So there's that to be aware of, but nevertheless, coverage really, coverage is the kind of biggest definer of the relative quality of your sequencing experiment in terms of the capturing of rare variation in particular. Transition-transversion ratio. So early in the days of sequencing, there was a bit of confusion about how to know whether or not you were right about something. It's like, well, we've got all this data, and not only do we have all of this data, we can make variant calls, but there are kind of two things going on. There's a kind of pathological set of consistent errors that these machines are throwing up that we don't want to kind of include in our analysis, as well as, a lot of real variation that we're identifying for the first time. So it's really hard to know how best to calibrate these things. And one of the observations that was made in the 1,000 Genomes project that's kind of particularly useful is if you think about the mutational space, okay? So if you think about, starting from a reference base of A, C, G, or T, you can then go to, if you're an A, you can go to C or G or T, okay? Now, you remember from your basic, biology, kind of bio 101 or whatever, we've got two kinds of DNA. We've got purines and pyrimidines, and the purines are AG and the pyrimidines are CT. And the little mnemonic device I was taught was purine is pure silver, AG is silver. You have to remember silver as well. I don't know if that helps anyone, but anyway. Nevertheless, so the kind of mutation within purine or within pyrimidine class is called a transition, okay? So that's called a transition, that's a TI. And then there's TV, there's transversions. Some people call transitions TSs, but I don't like to do that because they're both trans things. But anyway, so the transition is within DNA base type, so within purine or within pyrimidine. And then transversion is across type, so it's from pyrimidine to purine or from purine to pyrimidine, okay? Now, if mutation is a random process, you've got a TITV of 0.5. Okay, so you've got equally likely chance of being, you've got one shot at being a transition, two shots at being a transversion. That gives you a TITV of 0.5. Well, the mutational process in the human genome is not a random phenomenon. It's actually got some amount of structure to it. And the most noticeable place where we see this structure is that when we look at the kind of exome content, so if we look at those coding regions, we see a much higher TITV than we see at the genome at large. We actually see a TITV of around 3.25. When we look at the genome in general, so we look at all of the genome, we see a TITV of 2.25. So those kind of rough estimates of transition-transversion ratio is a pretty good proxy for the overall quality of the data set that you're having. And that's really what I'm trying to hammer home here, is this idea that the kinds of mutations that you're observing really help you figure out, how well your data are performing. Unfortunately, this isn't the, only way that you can kind of perfect things, because there are two ways of improving your TITV. One way is, removing false calls, and so that'll drop out your kind of 0.5 random expectation, since an error should be a random process and should have this TITV of 0.5. But the other way is to artificially boost the number of transitions that you call. And I have seen callers that do that as well. So do bear in mind that it's not a perfect. Yeah, Bill.
Okay. Sorry, don't mean to interrupt this focus.
Sure.
You may well come to this, and so dismiss it. But it relates to this coverage, because I agree there's ambiguity there, and many of us who are doing this understand the depth of reading a position. It's the percentage of the whole genome that now gets us worried about in terms of how much coverage. Is it 75, 85, 90%? And when you're coming up negative, this starts to become a really big issue. And a lot of people refer to that as coverage. So maybe
right. So what Bill is raising is a very legitimate secondary consideration. So there's coverage at a position, which is the defining characteristic of how well you identify variation at that position. But when you're talking about capturing an exome, there is coverage of the exome as well. So when we do our sequencing here, we have a kind of rough and ready rule of thumb that I think Mark just kind of offhandedly proposed in some meeting and has now become fiat. Funny how these things happen. That you basically need to achieve 80% of the exome at a 20x coverage and 90% of the exome at a 10x coverage. Now, to answer what I like to think of as the God of the gaps argument in the analysis of any sort of thing, Bill's right, we're missing somewhere between 10 and 20% of any given exome, maybe a little bit less. We've gotten a lot more sophisticated at kind of creating more uniform coverage across the entire exome, but it's still a very big place, so that causes problems. I would say our hit rate is not as high our hit rate, even if you take off the 10%, is still much lower than that. So I think it's probably a blend of both things, that we're getting some amount of difficulty in the identification of the region because we're just missing the place itself, and that there are some bits of the exome that we're not capturing properly. But at the same time, I think the kind of genetic architecture is probably stymieing us a lot more for a lot of the things that we haven't understood or appreciated. Or simple case number. If you're going to try and solve one of these rare Mendelian diseases and you only have one case, you've set yourself a very, very difficult task. If you have two cases, your task is actually much, much easier than it is just with a single case. So if you can kind of identify that clinically homogeneous severe phenotype, that really, really, really will help kind of identify. And most of those papers that I cited at the earlier days, some of them are one-off patient weirdnesses in genes that are already established, and that kind of helps you along. But a lot of the ones where we've made really novel discoveries are where you get 4 or 6 or 8 of these very rare diseases through the kind of global community of presentation of disease.
Is that a property of a physician or a run or both? In other words, is it centromeres are always missing, and no matter how many runs you get, you're never going to get those?
So yeah, sure. So the question is kind of what is the nature of this coverage, and the way the technology is working is not uniform with respect to, say, certain sequence contexts and so on and so forth. So 2 years ago, I looked at the kind of coverage across exome as a function of the GC content, and the more GC content that you had, so when you were very high in GC content, the worse off the coverage was a general rule. Now, there are also places where when GC content goes very low, that's sometimes weird, but that may have to do with lack of specificity of the sequence itself. So it's not independent what is being captured from experiment to experiment. There is variation in terms of that, but there is a correlational structure as well that's defined by the technology that you're using and also the protocols that are used, because they've tried to develop protocols to kind of more evenly cover the genome rather than the kind of variation as a consequence of the sequence context. So that's a moving target, but it's not simply that you're, a base that you capture very well in one experiment, you're much more likely to capture well in a subsequent experiment, and one that you don't capture very well at one experiment, you can solve this by really jacking up coverage, or if you have, like linkage information or a priori kind of idea of where to kind of go after explicitly, you can evaluate how well you've done. So the evaluation of how well you've done is also a kind of important component, I'd say. All right. So when we look at sequencing data, we look at a couple of different important things. The kind of number of copies of the allele that we observe in the data set is a useful shorthand. So when you're talking about your sequencing data set, people will talk about singletons. And usually what they mean by singletons is that I see one copy of that variant in my data set. Obviously, that singleton is a moving target as you increase your sample size. The fact, the stability of every singleton call is not perfect. Some singletons will now kind of transition to doubletons and so on and so forth. But the number of copies of an individual variant or non-reference allele in particular is worth flagging. The kind of case control counts, most of the experiments that are being done are case control. Sequencing on quantitative traits is lagging behind, as quantitative traits tend to lag behind of most of the kind of medically relevant phenotypes blazing the path forward. And then there's just a quick note on this mutational type. So when we look at the different kinds of mutations in a coding region, we have this synonymous stroke silent. So the idea here is that this is a base pair change in the exome that is creating the same amino acid. Now, these mutations sometimes are functionally relevant, sometimes, but very rarely. If you look at the selective pressure on synonymous mutations across the exome, it's very much weaker, almost non-existent and indistinguishable from intergenic regions. In contrast to these missense and nonsense mutations. So missenses are variants that are changing the coding sequence, but it's still being transcribed and potentially translated, but it may disrupt the protein, it may change the folding structure, it may do any number of nasty things biologically. And then there's this nonsense, which is this premature stop codon. So we've got that, the stop codons that say, all right, I'm done, I don't want any more protein, stop here. You can have mutations that introduce those into the coding region, and that'll really also sometimes cause problems. And then if you're really enthusiastic about learning a little bit more about nonsense mutations, Google nonsense-mediated decay, since there are some, stop mutations that once they occur, they are, they're transcribed, but they're not translated. The transcript is decayed away. So there's some part of the machinery that says, hey, that's a bit too early for that nonsense mutation. I'm uncomfortable with that. I'm not going to try and make this protein because that might cause a problem. Because a malformed protein hanging around the biological system may actually be more deleterious than the lack of one copy of that protein. So if you think about Huntington's, for example, that's a poorly formed protein, and that's causing problems. There's read length. So the length of the number of bases that you get at your core kind of basic raw output, your read, really does have an impact on how well you're going to be able to align the sequence, how well you're going to be capture, kind of the repeat sequences that are variable in the population, the mini satellites, the microsatellites, those sorts of repeat sequences. Longer reads means that you have a better job at really detecting and really calculating what the allele for an individual is at those positions. It also has an impact on, kind of smaller insertion and deletion mutations. So, the kind of bigger reads generally tend to perform a little bit better. Read length is more or less defined technologically based on how long quality persists. So basically, as you push those read lengths longer and longer, you start to get more garbage. And at a certain point, garbage, based data doesn't really help you understand anything about genetic variation. And so it's now effectively a waste of time and money to deal with those sorts of things. So that's really the kind of rate limiter in the read length approach in this day and age. Some technologies have much longer reads but much lower average quality, so there's a kind of push me, pull you on that level as well. There's this kind of mapping quality score idea and this base alignment quality score. And these there are lots of different quality scores that have been developed for sequencing analysis. And these quality scores come under different names and different guises and so on and so forth. But the mapping quality score is really an idea of how well this base really should be at this position in the genome, whereas the base, kind of alignment quality score is maybe a little bit over and above that that's for the base. The mapping quality score is for the read, sorry. So you get a whole read, how well this read is doing, and then how well this base is doing within the context of this read. And then there's the just general quality score of the base itself, and then you get quality scores for genotypes and so on and so forth. All of these things mean different things at different times because they kind of keep updating in terms of what we understand about the error processes and modeling the different sequencing technologies. So it's difficult to give you a kind of clear idea of exactly what is going on right now because it really won't persist. But there, you can kind of go and peruse GATK and Picard and so on and so forth. So those are the kind of, oh, that's interesting. What's that doing here? Anyway. All right, so those are the kind of core ideas. I guess the only other thing that I would highlight in particular is this step of quality calibration and annotation. So basically what we do is we take the sequence context, for example, and we use that to calculate how often we see a mismatch of the base internal to a specific sequence context. And that mismatch is a proxy for the overall, kind of error rate. Because in most places, in most everyone's exome or genome, you're going to match the reference. You have very few mutations in the scheme of things. And you've got a huge amount of data. I mean, we're really talking about millions and millions of bases. And so we can use this volume of data to kind of iteratively learn what the error properties of the individual sequencing data sets actually are. And that's kind of really the kind of insight that has kind of made this happen and made this a reality. If you want to learn more about that, I recommend you read the kind of DePristo et al. Manuscript that kind of outlines GATK. It goes through some of these concepts in much more detail than I have kind of time to manage now. And if you have any other questions, feel free to drop me an email or chat with me afterwards. So let's see, how am I doing on time? Good. We've got, so we get variant calls. So it goes through the sequencing pipeline. There's variant discovery and variant calling. That's what GATK is doing. And if you generate sequence data here, you will be delivered a VCF, which is a variant call file, which has all of the variation at the time at which, under best practices at this point in time, yada, 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 that will represent all of the genetic variation that they've identified from the sequencing experiment. Okay, so that is the deliverable. If you have sequencing done here, that's what you will get. That is not uniform for all purveyors of sequencing technology. Some places just say, here you go, here's your raw sequence data, have fun, good luck. And we have ways of analyzing that, and there's a fee for service for that here as well at the Broad, for your kind of awareness, is that you can say, I have gotten raw data from this technology from this place, and I would like to call it, and will you please do this for me and give me an idea, and they will do so. That's on the Broad price sheet and so on and so forth. So these are useful things, but let's talk a little bit about analysis, because I'm a statistical geneticist, and I spend a lot of time talking about these kinds of problems as well. So let's think about sequencing data. We've got this kind of generic kind of cartoon picture of a haplotype, and then these little orange points are genetic variants, just as a kind of very stylized representation of the data itself. And really, in ye olde days of, a year and a half ago, when we were doing lots of GWAS, or we're still doing lots of GWAS, we would do single locus associations. So we would just kind of look at a single point, and we would say, how many alleles in cases versus how many alleles in controls. You've already had the GWAS primer, so I won't kind of belabor this point too much. But that's really the way that we would approach most of the testing that we would do, is like, is this variant associated? Yes or no? All right, let's move on. Now, I think my thing is dying. So single locus association, we got lots of different ways of testing. You can do kind of regression or a t-test or ANOVA or so on and so forth. But there's a problem with all of these tests, is that if we have a very, very, very few number of alternate alleles at any position, then we won't actually be able to formally test anything and get anything approaching a sufficient p-value to write our Nature and Science papers that we're all excited to write. So few observations, our best significance is not actually that impressive. It's quite large. We want small. So if I've got a doubleton, 2 copies, and I got 10,000 cases and 10,000 controls, that would still be a large sequencing experiment right now. To do so, really, with only 2 copies, the best p-value I can get is basically a 0.5. And I can't do anything with 0.5. I don't know anything about this variant. I don't have enough copies to really tell what's going on. And we can explore the properties of this kind of problem a little bit further. We can look at, so this is a kind of pair of QQ plots. And what this is kind of 40 minor alleles in a sample of 2,000. And then this is 80 minor alleles in a sample of 10,000. And what we have is the minus log 10 p-value that we would expect based on the theoretical distribution under the uniformity. So we have a uniform distribution for, say, 1 billion such tests of 40 copies in 2,000 individuals. And then what we have on the y here is what we observe. And what I want to call your attention to is, I've done a couple of funny things with this. The first thing is that it's kind of got this step-like stagger thing going on. Well, that's the quantization. So there's basically one p-value for the configuration of 39 variants in one group versus one variant in the other group. That's going to give you a single p-value every time. So we'll see a number of copies of those, but that's okay, because that's what we would expect probabilistically. But the more important thing is that it's really skirting below the line. And this is really washed out. So it's skirting below the line, and there's a third line right here that's really following here. So what is this skirting below the line? Why am I stressing this? Why am I highlighting this? What this is saying is that the asymptotic properties, so the way that we expect our test to behave under the assumption of large sample sizes, it's not being met. We're seeing a deflation in the distribution of test statistics. So you can't just run a kind of single locus association test across your entire exome or genome and all that kind of stuff and expect it to conform to the uniform distribution for the p-value or the 1 degree of freedom chi-square distribution for the chi-square. The QQ plot, just based on your raw results, is not actually going to be informative to how well your overall distribution is inflated or deflated or all of those things. And that's because we're not achieving these asymptotic properties. We're not getting enough copies of the variant to really test it properly. Now, when we move to 80 variants, we're doing much better. We're much more on this kind of x equals y line. And so this dimensionality, this asymptotic properties of the test statistic are an important consideration when evaluating an experiment. at a single locus association level. Nevertheless, our overall problem is, well, we don't see enough copies of the kind of rare alleles or the rare, the alternate alleles in the data set to actually test them at an individual variant basis. We would like to do so, we like that, but it's not the best thing. So what's the solution? Well, let's lump things together. That sounds like a good idea. Not really rocket science at the end of the day. There have been a number of papers that do this sort of thing, but we'll go back to our little silly cartoon, and you can imagine a rare variant test of, in a region, do you have a rare variant, yes or no? Make that a dummy variable, put it into a regression, analyze whether having a rare variant predisposes to your phenotype. Or you could compare the rates of rare variants in your two groups, or any number of these different things. But basically, rare variant, yes or no, you can add a little twist, and you can say, well, I want to know how many rare variants you have, because I expect more rare variants or bad rare variants, or something like that. And these testing space has been explored in a number of different ways. I guess the kind of Lee and Leal, an American journal from a couple of years ago, goes through the kind of collapsing and combining method for evaluating a region for association. And basically what they do is they define some allele frequency threshold, and they say, below this allele frequency, we're going to collapse everything into a single rare variant, yes or no, or number of alleles in cases versus number of alleles in controls, so counting the number of rare variants. And then above that, they'll just do it in your old-fashioned single locus association way. So that's the kind of general concept. Well before Lee and Leal, there was the kind of Hobbs and Cohen work on PCSK9, which is kind of very important in lipid biology if you think about metabolics. And what they did was they basically developed this thing called the Cohort allelic sum test. And they conducted a burden test in the same way of case alleles versus control alleles. But the other twist that they added to it was to say, well, I think that most of my cases are going to have functionally relevant alleles. So the kinds of things that are missense and nonsense, those kinds of alleles are what I think we're going to see more signal in. And we can compare that against the synonymous mutation. So we can have a kind of background rate of synonymous mutation in cases versus controls, and then we'll see hopefully more functional alleles in cases versus controls, and that's what they see. Whether they develop that before looking at the data or after, I don't know, but it certainly has a biological motivation and logic to it that does help kind of characterize a little bit more of the lumped rare variants together and see what happens. And you just do a little chi-square test. You can kind of do your 2x2 table of cases, controls, synonymous, non-synonymous, or functional alleles or whatever. And there's your chi-square, nice and simple. And so here's a little bit more about the combined multivariate and collapsing. Again, it's build this regression, test the ones that you can test at a single variant level with single variants, test the rare variants all as a group together. But there's a little bit of problem with that, is that there's a lot of neutral variation in the genome. So you remember that slide from earlier in the talk where I was like, we got lots of nasty mutations in our genomes. Everyone, we're all going to die. So that is a kind of background rate of mutation that we're going to have to deal with. And that's really a bit of a challenge, because the more neutral variation you add to your test, the worse off your test is going to perform. So let us imagine a gene, just for a moment, take a moment, have a little thought experiment. Let us imagine a gene with 50 variants. All variants have 6 copies in my case control data set, okay? Now of those 50 variants, all got 6 copies, so we got overall in total 300 rare variants in our data set, okay? Of those, there are 3 that are really bad for you, okay? And 47 that are completely neutral. Well, if we have 3 that are really bad, and they're all in our cases, so we're going to make them, if you have one of these rare variants, you will be a case. Well, if I got a big case control, I get my 18 at the outset, and if I knew what 3 those were, my test statistic would be 18 to 0, and I'd be like, hey, that's pretty good. That's better than what most people are seeing in sequencing right now. But I don't know which the 3 are, so I've got the 3 that are giving me 18, and then the remaining 282 are equally divided between our 2 groups. And so I've got 141 added to each side, and so it's 141 in my controls plus, versus 159 in my cases. That's not so impressive. Not a very big odds ratio, not a very big chi-square. If you compare the chi-squares for those 2 scenarios, you're going to get very different answers. And it's really this neutral variation that's causing probably the most difficulty in the identification of what are really functionally relevant genes to the diseases that we're looking at. That in the biological architecture involving many, many genes in all likelihood. So there are a couple of ways of solving this particular problem. Kind of Madsen and Browning wrote a paper in PLOS Genetics that was like, hey, we can use functional information to try and weight things, but we can also use allele frequency information. So if we have the model that things that are causing disease or things that are influencing phenotype are biologically active, therefore if they're biologically active, they tend to be selected against. If they tend to be selected against, they tend to be rarer. Therefore, we can use allele frequency information to try and incorporate that into the test statistic. And so they talk about weighting with respect to the inverse of the allele frequency. So as the allele frequency goes down, the contribution of a rare variant goes up. Second to that, you can use functional weighting. So there's polyphen, polyphen2, SIFT, all kinds of different ways of annotating the coding region of the human genome. Those annotations come with quantitative scores. We can use those quantitative scores to potentially improve the power of our test. If we think something is very nasty, then maybe we should weight it a little bit more. Maybe weight a nonsense mutation more than a missense mutation, et cetera, et cetera, et cetera. And then, so that's kind of one approach and one set of approaches and one kind of class of thinking. You can go and read that paper in PLOS Genetics. It'll help you out in terms of characterizing those kinds of tests. But Alkas Price and Shamil Suniev and others here locally also hit upon the idea of this variable threshold idea. So maybe we're not so good at picking what rare should be. Maybe we don't really know where the kind of allele frequency selective coefficient is going to kick in. So why don't we just try them all? And to try them all, so there's Madsen Browning. There's a little bit more about the weights. But to try them all in variable threshold, what we do is we basically take every minor allele frequency threshold up to a given maximum minor allele frequency that you specify, maybe 5%, maybe 1%, whatever, something like that. And then you do this kind of case control, say, chi-square. So you do your rare variant regression test or your rare variant chi-square test or whatever. And what you're looking for is your maximum test statistic across all of the different thresholds that you use, okay? So you get your maximum test statistic and you're like, this is great. This is my maximum test statistic. I've got as much signal as I can possibly squeeze in at this point in time. The problem is the distribution of that test statistic is pretty unknowable. In the scheme of statistical distributions, that's a pretty ugly one. And because there's nasty correlational structure and all that kind of stuff. So what do we do when we don't know the distribution of a test statistic? Well, we use permutation. So you then permute case control labels and you say, you're a case now, you're a control now, whatever. You go through this exact same process, and go across the allele frequency spectrum, identify your maximum test statistic from that, and now you can generate an empirical distribution of maximal test statistics. And this empirical distribution of maximal test statistics is what you will use as your p-value calculator. So if I observe, like a chi-square of 7, I do 10,000 permutations of case control status, and the best kind of chi-square I observe from all of those permutations is 6.5. Then that means my p-value significance right now is 1 over 10,001. And there's a little bit of an arcane rule for that, but it doesn't really matter. But what this kind of shows to really underscore is that the best p-value that you can achieve under this kind of approach is really influenced by the number of permutations that you do. So if you only do 100 permutations, you're never going to have a p-value better than 1 in 100. It's just not going to happen. So that's kind of some of the computational considerations there. But compared to a sequencing pipeline, this is small potatoes. Okay, so we can think a little bit more about this. Well, maybe the biology is a little tricky. Maybe it's a little more complicated than that. Maybe there are regions of the genome where you have variation that increases your risk to something, and then variation that decreases your risk to something. Okay, so that is to say, there are a mixture of neutral, risk, and protective effects in the region. Well, what might that look like? So let's now go for a little coin tossing sidebar thought experiment. Okay, so thinking about coin tossing. Let us imagine 2 sets of coins, okay? The first set of coins we have is a biased coin. It has a 75% chance of coming up heads and a 25% chance of coming up tails, okay? And we can contrast that with a the second set of coins that's really a mixture of coins. So it's half of the coins always come up heads, and half of the coins are fair coins. There's 50-50 shot of heads versus tails. Okay, so we got our two groups of coins. If we do lots and lots and lots of coin tosses from group 1 or from group 2, we get the same expected number of heads. Okay, so we're still expecting basically a heads 3 out of 4 times on the biased coin and 3 out of 4 times on this 50% Mendelian coins that are double-sided head coins and 50% neutral coins. Well, what does that mean? Well, it means we get the same expectation. So here's our distribution of kind of heads out of tails out of 2 trials. But let's look at the marginals for a second. And when we see the mixed coin, what we see is we see a lot more 2 to 0s. Okay, so we see more 2 to 0s than the biased coin. But we also see more 0 to 2s. So we're seeing both of these observations. We're seeing a kind of movement away from that 1-to-1 observation of 1 heads and 1 tails in 2 trials. And so that's really a different pattern of data. Okay, so what we're talking about is a change in the probabilities of observing 2 heads and 2 tails in contrast between a mixed coin and a biased coin. So the problem now could be characterized as what are some of the ways we might go about trying to detect a mixture. Okay, so that's really what we're interested in identifying, is a mixture of this binomial process, because what we're thinking about, what we're conceptualizing, is that a rare variant that we observe in our study is assigned either to a case or to a control. If it's assigned to, if it's a risk variant, it's got a higher probability of being in a case. If it's a protective variant, it's got a higher probability of being in a control. And if it's a neutral variant, it's, equally likely in either group. So really that mixture is what we're interested in trying to test. So in 1966, Jerzy Neiman and Elizabeth Scott did some fancy maths. You can go and read the paper. It's like on composite likelihood testing. It's really, really great on optimal tests of composite hypotheses. Really lovely title. But basically what they do is they define a series of functions that are really well powered and well designed to capture mixtures. That's really the kind of name of the game, that we're trying to capture mixtures here. And then this has been kind of explicitly applied to a kind of binomial context of, say, case versus control by Zelterman and Chen in 1988. So we adopted this framework and wrote the kind of C alpha paper in PLOS Genetics that you can go and read about. Really, Catherine Rader did her PhD on these kinds of mixtures, and so she was a really good collaborator to have in testing for mixtures. So let's go back to our visual representation of the data. How would this look in a kind of C alpha conceptual level? Really, we've got our case count to control count, our singletons, doubletons, so on and so forth. And this is a kind of Christmas tree plot. So this is the probability of observing a kind of outcome depending on the number of copies. So up here, this would be 2 copies of the allele in cases, in the overall data set. And, a quarter of the time, if you've got equal numbers of cases and controls, a quarter of the time you'll see a 2 to 0, half the time you'll see a 1 to 1, and a quarter of the time you'll see a 0 to 2. And so on and so forth. And the pattern of data that we're really looking for is this over-dispersion, this excess variability, this piling up of mutations on the kind of outside of the Christmas tree. And that really does give us a different kind of information in contrast to a simple burden test where we just count up the number of alleles in cases versus the number of alleles in controls. We can apply this to real data. That really didn't render very well. Let's focus on ApoB, because ApoB was a nice kind of story, so we're not seeing the probabilities here. But what we are seeing is we see both a 6 to 0 and a 0 to 6 in a sample of kind of extremes. So you've got very high triglycerides and very low triglycerides, and we do sequencing in the region, and we scan ApoB, which we know to be involved in triglycerides. It's not like we're making a novel discovery here. But what we are observing is a pattern consistent with this idea of a mixture of things that may dramatically increase your triglycerides or dramatically decrease your triglycerides. So the kind of 6 to 0 or 0 to 6 in cases versus controls. There's another statistical approach that kind of does very similar things to C alpha under some parameterizations, but is really much more flexible, but has a very nice feature. So C alpha is a kind of closed form case control. You can't add any covariates or be fancy or anything like that. But there's this Skat. And there's some nasty equations up there, but what I really want to kind of impress upon you is not walk through all of the math per se, but it's really have this idea of this K. So this is a kernel-based statistic. Current trends in statistical analysis are in this kind of kernel-based approach. I mean, that's like modern statistics is focused on that amongst many other things, but this is the kind of flexible framework that you can use to test more or less anything in any way that you so choose. But this K is really basically a representation of the genotypic similarity in the region. And so really what this test is trying to do is say, if you guys are genetically similar across the rare variants in this region, then I expect you to be phenotypically similar. And let's try and test that. And that's really what what Skat is trying to do. But you can do it while including fixed effects of things like age and sex or principal components or whatever and so on and so forth. What about singletons? They're kind of a problem. It's difficult to think about that being a good binomial process of having more variability because you get one copy. So it's going to be in a case or a control. There really isn't a lot of opportunity otherwise. So, much of the variation that we're identifying from these kinds of activities are in fact, singletons. So when we aggregated 12,000 samples together for exome chip, over half of the missense mutations, nonsense mutations, and splice site mutations that we saw in that set of 12,000 individuals were singletons. One copy in 12,000 individuals. And that's, kind of like more than half of the 1.5 million variants we saw. So, it's kind of important and relevant there. So for C alpha, you can kind of collapse singletons down into a single meta singleton variant that you throw into like maybe a 30 copy allele or something and look at the rate in cases versus controls. Skat-O extends the variance component framework to kind of try and jointly test these things. So the idea is that you have a little bit of a burden contribution from the singletons in case versus controls, as well as this genetic similarity with phenotypic similarity engine going on the other side. There are a whole bunch of other methods that are out there. Really, this has been a very active area of research in the last couple of years in terms of people trying to define new and different tests. At the end of the day, they more or less break down into a couple of very kind of concrete camps. There are burden tests of, how many alleles in one group versus another. There are lots of ways of doing that. That's the core idea. There are these two-sided tests like C alpha and Skat that basically try and say, if you're more phenotypically similar, you should be more genetically similar, and we'll try and capture that information in a couple of different ways, but allow for effects that move in both directions. And then there's another test that Sean Purcell's implemented in PlinkSeq that's also relevant to this discussion, and it's what he calls the so-called unique test, which is the same as a burden test of how many variants do you see in cases versus controls, but it only counts variants that are unique to one group or another. For most of these tests, best practices still involve permutation to define the distribution of test statistics. The reason for that is you're still grappling with small counts. I mean, our sample sizes for sequencing still very, very small, so we really have to kind of get to much larger sample sizes before we might want to start to rely on some of the asymptotic properties of the test statistics that we might be using. So this concept of dimensionality that I kind of briefly mentioned, but we'll highlight again, and let's see, I got 3 minutes, so I'll probably end here. So this dimensionality of the exome, or why I want more variants, kind of a Dr. Strangelove title. So we do our C alpha kind of test. We're looking for over dispersion. We condition on between 2 and 40 kind of counts of the minor allele. And I'll show you a little bit of data based on 700 samples. The kinds of ways of thinking about this scale based on your sample size in general, so it's not specific to this 700. So here's our kind of QQ plot of gene tests, okay? Not very exciting, kind of nice in following x equals y. So we got our expected p-value, our observed minus log 10 p-value. No screaming signals, and that's, I would say, the general response from looking at 1,000 cases and 1,000 controls of autism at this point in time. But one of the things that we noticed relatively early on is in that same way that we looked at the single variant association, and we saw that deflation in the distribution, so things kind of running under that x equals y line. We also have the same sort of problem with testing genes. There are a bunch of genes where you only get like 2 variants, and 2 variants where you've got a combined number of alternate alleles of like 6. aren't really gonna, it's not really testable. You're not really gonna get anywhere in terms of identification of novel signal there, and you're also not gonna achieve those asymptotic properties. So you're not gonna have the best possible p-value you can have is not actually that particularly impressive. And so what Sean implemented in PlinkSeq, and I think this is a really valuable contribution, is this concept of ISTAT. And what ISTAT is doing is, so you do permutation just like we do for all these tests, and we like our permutation and so on and so forth. And once we've done permutation, for each permutation we track the number of times that we see exactly the same maximum test statistic, okay? So our largest test statistic, we grab those and we count the number of times that we see our largest test statistic, okay? And the reason that we do that is that the number of times you see that largest test statistic divided by the total number of tests that you do is the best possible p-value you can achieve for that gene. And so this is a histogram of the minus log 10 of the ISTAT, so what is the best possible p-value I can achieve for that gene or transcript, and the frequency here on the y-axis. As you can see, most things are reasonably well, kind of distributed and can get up to 10 to the minus 3, 10 to the minus 4 without any real challenge. But you do see this tail of things. So you see about 10% of the genes having a p-value of 10, basically 0.1 or greater being the maximum achievable p-value. So that's really kind of an important consideration when evaluating the overall distribution of test statistics and also evaluating how much more sample you might need to include to overcome these kinds of problems. So if I move back down to an ISTAT of, say, 10 to the minus 3, the distribution is changed. So now I'm only looking at tests that can have a 10 to the minus 3 p-value or better. And when I do that, I see that my QQ plot is now different. It's now rising off the line a little bit. I don't think there's anything there unequivocally, identifiably, but it does change the shape of the distribution of test statistics that you observe across the exome. And so really the number of variants that you have really dictates a lot about how well you will be able to test those variants. And I think with that, I will close since I'll kind of throw up some references, and I think we're at about the time. And if there are any other questions, let me know. I'll be around and can be reached by email as well. Thank you.